Good afternoon, everyone. Thank you so much for joining. This is the third webinar of a month-long weekly series highlighting our 2022 graduate research fellowships. My name is Tanya Torres, and I am California Sea Grant's research associate. My work primarily focuses on marine debris and trash pollution, but today I'm helping out hosting this webinar. And so for those of you who don't know, our program, California Sea Grant, is a federally funded and university affiliated organization that supports applied ocean and coastal science for all of California. And we do this through our research grants, fellowships, extension programming, and communications and outreach. And so today we're here to celebrate our graduate research fellows. The Graduate Research Fellowship is one type of the many fellowship opportunities that California Sea Grant provides. We provide other research, policy, and extension-related fellowships, and you can visit our website if you'd like to learn more about these opportunities. We have a fellowship page and a newsletter that you can sign up for, and I believe we currently have an opportunity out right now. Our Delta Science Research Fellowship is open, but I think the deadline is approaching very quickly. But I'll put those links in the chat for you. And so the Graduate Research Fellowship Program began back in 2020 to directly support graduate students working on applied research that meets California Sea Grant's strategic plan focus areas. And these are resilient coastal communities, economies, sustainable fisheries, and healthy coastal ecosystems. And so during the 2-year fellowship, the graduate research fellows are directly mentored by a research mentor and a community mentor, and they develop an outreach plan as part of their project to ensure that their research is useful for the entities and communities invested in their work. And so, like I said, throughout the month of June, we are proud to feature talks by all of our 2022 cohort of graduate research fellows, so they can share all the great work that they've been doing over the past 2 years. So a little plug is, this is the third one. We've got one more coming up next week. Tune in Tuesdays at 2 p.m. to learn about the rest of our graduate research fellows. The previous recordings from the first 2 weeks will be up on our website, probably after the month of June and July, and you can go back and look at any of the ones that you missed. But I will put the link to the schedule in the chat for you to also have. But that's it for my introduction. Today we have 3 presentations. We've got Genevieve Pugsley from the University of California, Santa Cruz. And we've got Brandon Quintana from Cal State University, Fullerton, and then Grace Auringer, sorry if I mispronounced that, from the University of California, Davis. And so we're going to have each presenter come on, share their screen, and give their presentation. We'll have time for Q&A after each presentation, and then we'll move on. But without further ado, I will pass it over to Eve.
Great. Hello. Yeah, let me just share my screen. Okay, awesome. Thank you for having me. My name is Eve Pugsley, and I'll be talking about my project today, which is called Development and Application of New Amino Acid Isotope Proxies to Reconstruct Central California Phytoplankton Community Composition. And, okay, sorry, my cat has decided it's a good time to walk on the screen. Okay. Okay, so first I'm just going to talk a little bit about how I got here. So I'm from Tacoma, Washington in the Pacific Northwest. And for college, I went out east to the College of William and Mary, where I majored in environmental geology. And while I was there, I was super lucky to get involved with a paleoclimate research lab. And we were studying using lake sediment cores, studying how changes in climate over the past several thousand years affected environmental changes and human civilizations. And so this research got me really interested in paleo-environmental reconstruction and how it can sort of inform us about interactions between climate and ecosystems. And then for my PhD, I came out to Santa Cruz, and my research focuses on how plankton ecosystems from coastal California have responded to past climate variations, specifically using deep sea coral and sedimentary archives. Okay, so today I'm talking about phytoplankton, which are microscopic marine algae. And not only are they beautiful, they're also globally important as major contributors to biogeochemical cycles of oxygen and carbon. And they also form the base of most marine food webs. And in coastal regions, specifically, different important phytoplankton functional groups, such as diatoms and dinoflagellates, play unique and different ecological roles. So it's important to understand sort of drivers of community composition shifts. And in addition to all the wonderful things that phytoplankton bring us, they can also cause problems. So harmful algal blooms present costly and dangerous challenges to coastal communities, including in California. And what we know, so the region that I'm focused on for this project is Central California. And we know from past work, and these are a few publications by Fisher et al. And Barth et al. From 2020, we know that phytoplankton community composition is closely tied to low frequency modes of climate variability, like the North Pacific Gyre Oscillation and the Pacific Decadal Oscillation. And understanding the drivers of these shifts is important for predicting future changes under anthropogenic climate variability and change. And so reconstructions of past phytoplankton community ecology can help us better understand sort of long-term relationships between climate and marine ecosystems. Okay, so the focus of my project is the essential amino acid carbon isotope fingerprinting proxy. And what I'm showing here is a eukaryotic tree of life, and circled are four of the sort of major phytoplankton groups. And you can see there's a lot of phylogenetic diversity. And with that phylogenetic diversity also comes metabolic diversity. And in particular, each of these groups create amino acids that have unique carbon isotope patterns relative to each other. And so we can use this as a proxy for community composition, and it has several advantages over other types of proxy in that it's applicable to all taxa, whether or not they sort of preserve hard part microfossils. So a lot of community composition estimates rely on preservation of microfossils in sediments, but all phytoplankton produce amino acids, even if they don't produce a shell. So it's useful in that way. This proxy is also independent of growth conditions. And what's really interesting is the essential amino acids, their isotopic signatures are transferred without alteration through food webs. So there's also a really cool potential to trace flows of carbon from primary producers through consumers and understand flows of carbon through food webs. So in the past, this proxy has only really been used to differentiate very distantly related taxa, such as cyanobacteria versus eukaryotic phytoplankton. And additionally, it was only previously demonstrated in culture and never tested on a mixed natural phytoplankton assemblage. So for my project, I had a first part where I was testing this new proxy on natural phytoplankton assemblages. And in the second part, involves applying the proxy to paleoceanographic archives from the Central California coast. So the methods for part one, I went out every other week for a year to the Santa Cruz Wharf. And along with undergraduate research volunteers, we collected water, brought it back to the lab and filtered it. And we ended up with something that looks like this, which are particle samples on a filter. And then I analyzed these particle samples for bulk carbon and nitrogen isotopes, as well as compound-specific isotopes of amino acids. And then I created training data sets from unialgal, meaning like one species of phytoplankton, lab cultures. And I created new lab cultures for this. And I also compiled data from the literature. And then we compared this lab culture training data to the results from the WRF particle samples. And then finally assessed the results against independent community composition estimates from a microscopy approach. And to get these microscopy estimates, we partnered with the Rafe Kudella's Biological Oceanography Lab at UC Santa Cruz. And they're a member of California HabMap, who monitors harmful algal blooms throughout the state of California. And they do weekly phytoplankton counts at the Santa Cruz Wharf. And here I'm just showing some of that data. So time is on the x-axis, and then the colors represent the relative abundance of diatoms in green and dinoflagellates in the pink color. And in Monterey Bay, there's a really strong, dramatic seasonal succession from diatom dominance in the spring and early summer to dinoflagellate dominance in the late summer and fall. And so this sort of created an natural laboratory experiment setting in which to do this project. Oh, and I should say on this figure, the X's represent days that I sampled at the wharf. Kind of on the top there. Okay. So first, I'm going to show some results from the training data, and I know there's a lot going on here. So just to orient you, each of the different training data sets, including MindNew data and literature data, is represented by a different shape. And the colors represent different taxonomic types. So diatoms are in green, dinoflagellates are in pink, and bacteria are in the kind of brownish yellow color. And what we found is that across all of the training data sets, that the most diagnostic amino acids for differentiating the two dominant phytoplankton types in Monterey Bay, which are diatoms and dinoflagellates, were valine and leucine. In particular, leucine and valine carbon isotope values tended to be higher in diatoms than in dinoflagellates. And also, I'll just point out that the wharf particle samples are plotted here in the gray color, and they fall between the eukaryotic algae and bacteria. And it'll become clear why that's important in the next couple slides. Okay, so now we're going to compare these training data sets to the data from my Monterey Bay particles. So in the circles, I'm showing the lab culture training data sets with, again, pink is dinoflagellates, green is diatoms, and the brown color is bacteria. And the triangles are the particle samples. And the colors here, so like the dark green color represents days when the phytoplankton community was dominated by like almost 100% diatoms. And the pink colors mean it was the phytoplankton community was dominated by almost 100% dinoflagellates. And what we find is that consistent with the training data sets, on days when diatoms were abundant, the valine and leucine values of those particles was higher, and vice versa. When dinoflagellates were abundant, we saw lower values for valine and leucine. And so this is really promising and shows that the proxy in some way is working as we expect. But we notice there's this offset where the valine and leucine values are higher overall than the eukaryotic algae and sort of pulled in the direction of that bacterial end member. So that suggests that in our particle samples, there was a major component of bacteria that was sort of muddying up the community composition signal. Okay, and then the question is, can we make quantitative community composition estimates using amino acid carbon isotope fingerprints? And this was sort of the first check on this using natural phytoplankton assemblages. So to do this, I used a Bayesian end member mixing R package called MixSciR to estimate the phytoplankton community composition of diatoms, dinoflagellates, and bacteria. And yeah, so what we find is that the predicted and actual percentage of dinoflagellates, we see some agreement in general with a correlation coefficient of 0.65. However, there's a greatly reduced range of variability in the predictions and large uncertainties. And I'll just point out on this graph that, so like the actual percent of dinoflagellates in the phytoplankton community ranges from 0 to 100, but the mixing model is only really predicting changes of around 20%. So we're not seeing sort of the full range of variability that we would hope to see from this proxy. So in conclusion from this part of the talk, the amino acid carbon isotope fingerprinting proxy does show promise for characterizing natural coastal phytoplankton assemblages. However, it seems like there's large isotopic signals coming from heterotrophic bacteria that are sort of complicating interpretation of this data and preventing us from getting fully quantitative community composition estimates using this proxy. And then for part 2 of my project involved application of this new proxy type to paleoarchives. And so I'll just go over some of briefly the initial goals and status of this part of the project. So originally I planned to apply this proxy to deep sea coral from Sir Ridge to better understand decadal scale variations in phytoplankton community composition. And we did complete the bulk and compound specific isotope analyses on several records from Sir Ridge. And we did learn a lot from different amino acid isotope proxies, but we found that the valine isotopic signal was poorly preserved in the coral. And as a result, we weren't really able to differentiate diatoms and dinoflagellates using this approach. And then the second way I'm applying this proxy is to sediment core records from Monterey Bay, with a goal to understand centennial to millennial scale changes in the phytoplankton community. And this is something I'm doing with my community mentor at USGS, Jason Addison. And he's working on a project to characterize productivity and phytoplankton changes through time all across the North American margin. And for this, I'm targeting the medieval climate anomaly, which is a warm period that occurred about 1,000 years ago that serves as a useful analog for future warming and drought in California. And this part of the project is in progress, but I'm really excited about it because they have this really rich multi-proxy data set that includes microfossil counts of harmful algal bloom species like Pseudonychia. And we're hoping that we can use information from the amino acid proxies to help better understand species without microfossils like dinoflagellates. Okay. And then what's next for me? I'm defending my PhD in spring 2025, and I'm sort of looking out now for what's next and applying to jobs, but I'm targeting a career as a coastal oceanographer at a university or government agency. And I want to thank all my funding sources, and especially California Sea Grant, obviously, for the awesome opportunity to do this project, as well as my faculty advisor, Matt McCarthy, our lab managers, Jeff Christensen, who helped me a lot with the lab culturing, especially my USGS community mentor, Jason Addison, 2 undergraduate researchers that helped me out, Haron Hyla and Chris Avila, and Rafe and Kendra from the Kudella lab, who provided the microscopy community composition estimates you saw, and also helped me with the culturing part of this project. And thank you for listening, and I'll take any questions.
Thank you, Eve. If anyone has any questions, please feel free to either raise your virtual hand through the Zoom platform, or you can feel free to come off of mute and ask your question. You can also add that to the chat. Does anybody have any questions for Eve? Well, maybe while we let people think, Eve, I had a question for you. Could you share maybe what was your biggest challenge, I would say, in doing this project? And I guess how did you kind of overcome that, or maybe there weren't any, but I was curious, if you had any like setbacks or anything.
I guess one big challenge I had was sort of logistical. So I didn't know how to culture phytoplankton before starting this project. So there was sort of a learning curve for me in doing that. And then I had a lot of issues with like the storms that happened my cat's here. Like landslides covering my road, and I couldn't get to my cultures. And so that was kind of hard, but and then obviously like the data didn't come out as like perfect as I'd hoped it was, but I think it's still an interesting story and we can learn from it. So
Awesome, thank you.
Thank you.
And if anyone else has any questions, feel free to add them to the chat or reach out to Eve. Eve will be on for the remainder of the webinar. Thank you, Eve. So next up, we'll have Brandon Quintana. Feel free to turn your camera on and you may share your screen. Brandon will be giving a presentation called Friend or Foe, Effects of Eelgrass Density on Filter Feeder Biomass and Condition Index in a Multi-Habitat Living Shoreline.
Cool.
So take it away, Brandon.
Yeah, so thanks everyone for coming. I'm going to start with like more of the background and then get into the research. So I'm Brandon Quintana, and I grew up in the underfunded part of Southeast Los Angeles, where I experienced environmental injustice from freeway pollution, which led to me developing asthma at an early age. And this experience with social inequity opened my eyes to the other injustices that historically excluded communities face. There was a lack of green spaces and finances here, which made it physically difficult to connect with nature. But I connected with nature in other ways, like watching Nat Geo documentaries with David Attenborough, or playing around in an inflatable pool and trying to stay afloat in 1 foot of water. I was finally able to immerse myself in nature through field research despite several hurdles. When I first began my marine science career, I felt very isolated because everywhere I looked around, I was usually the only person of color on the field stations. So now I try to maintain a welcoming space in conservation. At Cal State Fullerton, my efforts are focused on coastal restoration, particularly the living shoreline seen here with restored oysters and eelgrass, which I'll talk about more later, and what my 2022 graduate research fellowship was focused on. And I take an interdisciplinary approach on this work to try to support communities like mine by integrating environmental justice, education, and policy into this coastal work. And so our living shoreline is a great setting to learn about ocean sciences, and I work as education staff with Orange County Coastkeeper to help get underserved schools to learn about marine science. And I've held multiple symposiums that highlight people who have been historically excluded to integrate environmental justice frameworks into science and living shorelines. And we bring in policymakers like state senators and assembly members to support marine science restoration from the bills that they're supporting. And my connection with building community for marginalized groups and my own identity have shaped my work, and I'm really proud that through persistence, I've been able to overcome multiple barriers. And now in my role as a scientist and advocate, I need to contextualize my coastal work with a strong goal to achieve social equity. And it will require me to continue integrating principles like these to be my own version of David Attenborough, to mentor and inspire people who have intersecting identities like mine. So now to get into the research part. So we're going to talk about friend or foe, effective eelgrass on filter feeder biomass in a multi-habitat living shoreline. So we've all heard of the term slippery slope, and here we can see a slippery slope of riprap. They're one of the man-made coastal infrastructures made to combat coastal degradation, but at what cost? They might keep homes protected for now, but in the long term, they break down while increasing erosion and decreasing habitat. And there could be a potential solution along shallow sloping shorelines such as these, which is multi-habitat living shorelines that are made up of natural resources and placed strategically to decrease erosion and increase habitat. Target species of living shoreline projects are foundational species, which create and maintain habitat for other species. And if more than one foundation species is used in a living shoreline, as seen here, it is a multi-habitat living shoreline. Previous studies have shown that multi-habitat living shoreline projects have more restoration benefits than any single habitat. And for our purposes, we're going to be focusing on oysters and eelgrass. In our living shoreline configuration in Newport Bay, eelgrass decreases water velocity coming from downshore and captures sediment, which can increase shoreline resiliency, but the impact sedimentation has on oysters, mussels, and snails that also recruit to oyster bed habitat and are broadly classified as filter feeders depends on where the sediment is deposited. If shell accumulation is exceeded by sedimentation rates, it could be detrimental because it can obstruct the gills in oysters and other filter feeders. A multi-habitat living shoreline was created in Upper Newport Bay in 2016, including restored oysters and eelgrass, in order to assess whether Olympia oysters and other filter feeders perform better or worse with or without restored eelgrass. I will be comparing the biomass and condition, but we'll only be talking about biomass for this talk for time, of native and non-native filter feeders 1 year, 2018, and 4 years, 2021 post-restoration. And the question that we're asking is, filter feeder biomass affected by eelgrass-induced sedimentation. So what is biomass? It's made up of 2 questions: who is there and how much of them are there? Total biomass provides an estimate of how much ecosystem services will be returned. For instance, the higher the total biomass, the more filtration services can be provided by filter feeders. Here is a closer look at one of our restored oyster beds, and you can see that it has a higher total biomass to provide more filtration services per unit area versus the sparse oyster bed, which has a lower total biomass and is lacking its filtration services per unit area. So these are our predictions and hypotheses. So we predicted that when oyster beds and eelgrass are restored together, that we're going to see higher levels of sedimentation rates. And for our second and third, that when eelgrass is present or when eelgrass density is increasing, we're going to see a decrease in total biomass. And this is where the filter feeders were collected from and where Newport Bay is located. So they were collected from PCH, Pacific Coast Highway, Westcliff, De Anza, and Shellmaker. And this is the experimental design that's present at each of the site, and they're randomized in a different arrangement and consist of oysters stored alone, eelgrass are stored alone, a control with no manipulation, and oysters and eelgrass are stored in combination with each other. And since the project initiation, each site has experienced differential restoration and created a mosaic of restored habitat with varying densities of the two target species. So some of the oyster treatments have some eelgrass now, and some of the eelgrass has died back at the oyster eelgrass treatment. And this will be especially relevant in 2021. But we're only really concerned with the shellfish habitat, so we're going to be considering only the oyster treatment and the oyster eelgrass treatment. And these are the top 5 filter feeders that we found in our studies, which are Olerita, Adula species, C. spinosum, M. galloprovincialis, and M. synhousia. And I'm going to be using the asterisk to depict between native and non-native species. So the top 3 are native and the bottom 2 are non-native. And for sedimentation, it was assessed through mud deposition directly onto the oyster bed. And at each site, we used a point contact quadrat method by putting down a point contact quadrat onto the mud and using a graduated periodontal probe to measure the depth in millimeters. And for eelgrass density, it has been collected non-destructively every summer since 2016 using quadrat and transect techniques. And for excavations, that's how we get our filter feeders. So we put down n equals 10 quadrats down, then we clean off all the mud and other debris off. And then we sort the filter feeders by species. And then we can get to the biomass and condition index, where we weigh for total shell and tissue wet weight, seen separated here, and put it in a vacuum oven, seen here, for 22 hours at 100 degrees Celsius. And then we're able to weigh for the shell and tissue dry weight and quantify total biomass and per capita condition index. And we processed over 10,000 samples between the 2 years. So for 2018 sedimentation, 1 year post-restoration, we have on the x-axis site, and we only have 3 out of the 4 sites because for Shellmaker, there's no difference between the oyster and eelgrass treatment because there's no eelgrass at the oyster eelgrass treatment. And on the y-axis, we have mud deposition on the oyster bed in millimeters, and the green bar is the oyster treatment, and the brown bar is the oyster eelgrass treatment. So we assumed that eelgrass was going to impact oysters by potentially depositing sediment. But what did our measurements show? It showed that mud deposition onto oyster beds was not impacted by eelgrass, except at PCH, where deposition was higher, where oyster beds are restored alone. And, And in fact, mud deposition on oyster beds was generally not impacted by eelgrass, completely contrary to my hypothesis. Any negative impact is not being caused by sedimentation, as far as we can tell. And then looking at this from the perspective of eelgrass density, where we can include Shellmaker, and we have eelgrass density on the x-axis and mud deposition again on the y-axis, we saw that there was no effect of eelgrass density on sedimentation. And then fast forwarding, we again see that eelgrass density has no effect on sedimentation 4 years later. So not so much about how much eelgrass is there, but maybe something going on with the presence. And moving forward to biomass, we have species on the x-axis with the top 5 filter feeders and total biomass on the y-axis and the same green and brown bars. And what we found was that Olerita and M. synhousia had 3 times and 2 times higher biomass on oyster beds restored alone versus with eelgrass. So then looking at it from eelgrass density, we see that there is no impact of eelgrass density on total biomass. So it's not so much about how much eelgrass is there, but rather the presence of eelgrass that is somehow negatively affecting filter feeder biomass. And then when we fast forward to 2021, we are only looking at eelgrass density. And what we found was that there was no effect of eelgrass density on total biomass. So again, something about the presence, not so much how much is there. And this is kind of recapping all of the results, since it was kind of a lot of grass. And we see that for density, there's no effects onto the filter feeders, but then for a native Olerita and non-native M. synhousia, there was a negative effect of eelgrass presence on the biomass. And for eelgrass, it does not increase sedimentation onto restored oyster beds. And kind of getting into maybe reasoning for why, when we looked into the literature, we found that deposition rates were generally higher within seagrass beds versus outside of them. So maybe the seagrass is capturing the sediment and acting as a filter before it settles to the oyster bed, and suggests that we need a further investigation into fine-scale sedimentation patterns in eelgrass and adjacent oyster beds. And then for biomass, we were thinking kind of the same thing when it came to like the sediment, where maybe that's the case for the food. So in the literature, it said eelgrass presents reduced filter feeder food sources. And also, when you add in the density and patchiness, it can alter the hydrodynamics in a way that could reduce chlorophyll content by 50%. So we want to explore the connection between phytoplankton biomass and filter feeder biomass in the future. And kind of taking a step back to think about what the purpose of the co-restoration is, we need to think about in the context of this project, we were looking at biomass as a proxy for filtration function. And as we saw, it was kind of a negative impact from the eelgrass presence. And we would think maybe we don't want to restore or co-restore together. But other research in the lab that has looked at how co-restoration can increase overall biodiversity, eelgrass growth, and sedimentation while decreasing wave energy, which suggests that overall, we do want to co-restore oysters and eelgrass together. And significance for this project is kind of, now that we understand what the relationships between biomass are with these different factors, it can immediately inform co-restoration efforts and answer the question, are oysters and eelgrass friends or foes? And well, the answer is kind of complicated, and it really depends on what services or the goals of your restoration project are. And once those goals are decided on, then we can better restore species and habitat, and ultimately better protect species or ultimately better protect coastal communities living and depending on our coastline, which is particularly important in the face of climate change. And I would like to thank the Zacro Lab and California Sea Grant for their support. And here's my literature cited. And as for next steps, right now I've been at the California Coastal Commission as part of the California Sea Grant State Fellowship since January. And the different projects that I'm working on include working on affordable housing, integrating environmental justice into sea level rise guidance for the state, expanding nature-based solutions for the state, managing a lot of coastal development permits, working on state level coastal legislation, and federal level integration of renewable energy like offshore wind. And yeah, I'll take any questions. Thanks.
Hi, Brandon. Thank you. Excellent presentation. Does anybody have any questions for Brandon? Yes, we have Sarah. Would you like to just come off of mute and ask your question?
Hi, yeah, I was just curious, like how, if you could talk about how, like what some of the education outreach that you've done, like that you've performed in relation to your work. Sorry, I'm looking at my notes because I was writing this as you were talking. Like thinking about like with the sea level rise, affordable housing, those that are more at risk, how do you present a topic, like topics like that without causing a sense of fear or whatnot, but also empowering, in that, and then kind of tied to that separately is also like something like talking about, the presence of eelgrass affecting filter feeders. How do you explain that and make it accessible to somebody who, like, you were saying, like, didn't have access to checking out seagrass beds all the time? I'm just curious what you, if you could talk about that a little.
Yeah, so as far as our education efforts for this project, we did a Cal EPA grant where we were looking at kind of Santa Ana High School, where we were able to kind of help them come to Newport Bay where they hadn't been there before. So kind of place them there and then have them feel empowered in that way. Because we asked them, like, has anyone been here? And then no one raised their hand. So that's one of the parts that I left out for time. But we kind of helped them understand what was going on in Newport Bay, asked them what they thought was interesting, and then kind of helped like guide those projects a little bit forward to then coming up with like a poster and then thinking about how to present that and stuff. And then as far as like other like education, we do like an oyster shell string event too, where it's like the general public, where we kind of like put like all the oysters on a string, and then we kind of like have them hang it on like the side of their boat for 6 months throughout like their reproduction period. And then at the end, like it's kind of crazy how people become attached to them. Like one of the guys was returning his oyster shells, and he was like, I don't want to give these away. These are like my kids. And we're like, no, they're gonna go back to the ocean. We're gonna put it back into the living shoreline. And then he came back again this year being like, I'm so excited to take a picture this time before like my children come in for this year. So I think maybe just kind of what I try to do is like center the person and think about what's important to them and put it in a way where can I take out whatever like complicated topics or like words and put it into like layman's terms to think about, okay, I'm meeting you at like the level of information that you know based off of what I've asked, and think about what's important to you, and then that way you can keep on being engaged with it but feel like you can have something to say as well versus being like kind of cut off from that, whether it's like the wording or kind of like how it's presented to them, not so much like being like, hey, here's this living shoreline pamphlet and take it and whatever, but like kind of like, hey, like how's your morning, or how are you doing, have you been here before, or do you know, like kind of like stuff like that, like gauging what their level is and then going from there, I think is probably the best approach, and then thinking about how to like simplify it as best as you can.
That makes sense.
Yeah,
Yeah. No, sorry, I didn't mean to cut you off, but no, that's great. That's
yeah.
really sweet about the oyster shells. They were like, they're my food.
Yeah, I ended up taking a picture with him this time, so it was nice. But yeah, same thing for like the affordable housing. Like we're thinking, how do we say coastal development permit or like local coastal program? We just say coastal planning document or like something like more accessible, instead of like the words that we think are so like easy to know. Maybe it isn't as accessible for other people.
No, totally. Thank you. And feel free if you wanted to share any images like that. Sorry, I work with California Sea Grant. I probably should have said that. I'm the reporting and evaluation coordinator, and always trying to help with comm stuff. So if you have any photos that you wanted to share that feel free to share them with us. You know,
Mhm.
anyway,
Yeah.
that's all. But thank you so much.
Yeah, I'll look for the pictures. Thank you. Richard?
Hi. I was interested in knowing, have you correlated any ocean acidification data with your oysters and your eelgrass um studies that you've done? The reason why I'm asking is we have in Morro Bay, we started a Morro Bay estuary air monitoring project where you measure atmospheric carbon dioxide and give that information to our local researchers for measuring ocean acidification. And is that a variable that you're correlating with some of your studies with oysters and eelgrass densities?
I don't think right now we have like really strong ocean acidification data. It is like one of the points that we make when we're doing like the outreach efforts, like kind of like as like the waves are increasing, that can decrease like the hydrogen ions and like make the shells a little bit weaker to like predation and stuff. We like do like some like water quality measurements, like nitrogen, um, what else? Ammonium, but I don't think we've done like direct acidification of the shell yet. I can look into it and ask my advisor if we have anything about it, but I don't think we have like in-depth stuff about acidification, but really cool stuff about Morro Bay.
Thank you.
Thank you.
Right, thank you. Did anybody else have any questions for Brandon? All right, if not, feel free, if anything comes up, add it to the chat. Brandon will be around. We can move on to our last but not least presentation. We've got Grace Auringer from the University of California, Davis, going to give a presentation on Lamprey conservation genomics. So I will have Grace share screen. All right, and we see it. Take it away, Grace. Oh, Grace, I think
Perfect.
you are on mute. Okay, there you go.
Thank you. I wanted to kick it off with this great photo of Pacific lamprey. They are spectacular looking, in my opinion. So I'm here to talk to you about lampreys in California. I recently published a paper on mitochondrial phylogenetic analyses, and we found some really cool stuff, including previously unrecognized diversity. A little bit about my background. I studied biology at the University of Utah. There I developed an interest in genetics, and particularly as it relates to medicine. So I ended up working in the biotech field for a little bit at a company that focused on genetic-based medical tests. And then I had a really large pivot in my career, and I was able to get a job doing some field work, specifically rangeland vegetation surveys, but this was my pivot into ecology and conservation. And I was fortunate enough to then be able to combine these interests in my work at the Genomic Variation Laboratory. I was initially a laboratory technician, but I liked my work so much that I decided to pursue a PhD with the lab in integrative genetics and genomics at UC Davis. So why should we care about lampreys in California? They're a really understudied and underappreciated group of fish, but they contribute a lot ecologically. During the first part of their life, they burrow into the sediment, as seen in this picture here, and they participate in nutrient cycling, they maintain stream bed conditions, improve water quality, and especially as they get a little bit bigger and mature into the other life phases, they're a valuable prey item, and the anadromous forms of lamprey help deliver marine-derived nutrients upstream, which are super important to riparian habitats and freshwater systems. Additionally, they are a subsistence food and cultural resource for many different indigenous peoples across the west coast of North America. And they're this really ancient lineage with unique research potential in evolutionary biology and biomedical science. They appeared in the fossil record about 450 million years ago, which this timeline is not to scale, but was long before the dinosaurs roamed the earth. And their place as some of the most basal vertebrates have really interesting applications across a wide variety of fields of research. In California, we see a lot of barriers to lamprey conservation and management. We have limited knowledge about their diversity, abundance, and distribution. We also have a lack of awareness both in the broader community as well as across many management entities. There's inadequate historical data that is geographically and temporally fragmented, and when we do have data, it's not species specific, so it's somewhat incomplete. Lastly, there's taxonomic uncertainty. There's a lot of questions surrounding species boundaries in the lamprey lineage, and we see life history variation, and there's no real consensus on how to parse that variation taxonomically. So before we can really study lamprey to any degree, we first have to characterize what's present. So the overarching project goal was to characterize lamprey diversity. And the first way we wanted to do this was to identify the currently recognized species present at each sampled location using genetic species ID, and we utilize a method called DNA barcoding to do this. And this is basically taking a lamprey of unknown species, extracting DNA, sequencing a gene, and then comparing that to a public database, which allows us to then identify what species was present. Secondly, we wanted to compare the current taxonomy to the observed genetically distinct lineages. So to do this, we estimated molecular phylogenetic trees and performed species delimitation analyses. And a third aim of this project was to determine how this diversity that we've learned about is distributed across the landscape. Is it kind of scattered randomly, or do we see pockets of diversity in specific locations? So jumping into this first aim, well, actually, I'll back up and tell you a little bit about how we got our samples. We were fortunate enough to collaborate with ongoing monitoring programs across the state who helped participate in opportunistic sampling. And we were really excited that we were able to get lamprey samples from 19 sites across 15 subbasins. They were mostly larval individuals. And as you can see in this picture, there are not a lot of distinguishing features during this larval phase. So it's not surprising that over 50% of the samples we received were unidentified, even to the genus level. So once we had our samples, we extracted DNA and sequenced the cytochrome B gene. You don't really need to worry about that specific gene. It is a gene widely used in phylogenetic studies, and this specific gene had a lot of data and a lot of sequence data in public databases. So that enabled us to make comparisons to other populations that have been studied before that we weren't able to get DNA from. So to give you an idea of what we expected to see, these are the 8 species that are currently recognized, the species of lamprey known to occur across our study area. We see diversity in migration, feeding type, so we see parasitic and non-parasitic lampreys, and they are all of conservation concern of varying degrees. These 2 species that I've grayed out, we have so little information about, and we don't have any publicly available sequence data, so we're going to mostly focus on these other 6 species. To give you a better visual of what these 6 species look like, we see 3 species of migratory lamprey. The Pacific lamprey is kind of the superstar, the most well-known species of lamprey. It's also the biggest. We have the Klamath River lamprey and the Western River lamprey, which are also migratory. And then we see freshwater resident brook lamprey. These are freshwater resident and also tend to be non-parasitic. So to get at our first question, we wanted to know what species are present at each sampled site. So we used a method called DNA barcoding. And that method employs or really harnesses the power of these mismatches in DNA sequences. So once you have a long enough string of DNA, you start to see a pattern of where mismatches occur. And then if you have a really long sequence for several individuals, they start to look like a barcode. And just like you'd scan a barcode in a store, we can pipe that DNA barcode into the public database and compare to all the other sequences, and we are able to identify what species we have. So within the Lampetra genus, we're working with 2 different genera here, but first the Lampetra genus. There are 2 different species ranges shown here in yellow and in kind of the salmon color. So that's where we would expect to see Western River lamprey in yellow and Curran Brook lamprey in salmon. And then the black dots are sites where we did observe Western River lamprey, and the green dots represent sites where, or site, singular, the site where we found Curran Brook lamprey. And in the Entus venus genus, we've got 3 different species that we're looking at here, and the most wide-ranging is the Pacific lamprey shown in green. The black dots are sites where we found Pacific lamprey. The yellow sites are where we found the pit Klamath Brook lamprey, and then the dots in purple with a thick black ring are sites where we found both Pacific lamprey and Klamath River lamprey. So to summarize those results, we found Pacific lamprey at 14 sites. So it was definitely the most wide-ranging and the most abundant in our sample set. Followed by the Western River lamprey, the other migratory species, that was found at 7 sites. And then the other 3 species that we found were, as expected, found in more isolated regions across the state. So secondly, we wanted to look at the current taxonomy and how that fell out on a phylogenetic tree. This is very small, and I don't want you to really take anything away fine scale, but we can see that clearly at the genus level, we are correctly classifying the diversity. So all individuals that were classified as Lampetra are forming one clade, and all the Entosphenus are forming another. Another thing that I want to highlight here is just how different the shape of the trees are. So Entosphenus doesn't really have any long branches. This indicates that all of the individuals in that genus are very highly similar genetically. Whereas in Lampetra, we do see these really divergent branches. So we're looking at a lot more genetic diversity within that genus. This result is replicated in a different measure. This is genetic distance, which really just indicates how genetically differentiated lineages are. And so within Lampetra, we see these moderate genetic distance values in orange and salmon, versus in Entosphenus, everything is quite low. Everything's this even shade of yellow, indicating very little genetic differentiation. And of course, as expected, we see really, really high genetic distance values between these two genera. So what we've learned so far is that in Lampetra, we see really high genetic differentiation, and in Entosphenus, we see low genetic differentiation. So moving on to the Lampetra tree by itself, I do want to point out that these phylogenetic trees include publicly available sequences from previous studies, primarily Boguski et al. 2012. This is still a bit hard to look at, so I will make it a little simpler on the next slide, but what I want to highlight here is our molecular species delimitation results. So based on this data and this tree, we were able to delimit distinct species. And here, a unique color is a distinct species. We used multiple methods, and the most highly supported outputs are shown here. Now I'm going to make it a little bit easier to look at. We first have Western river lamprey, and this species is actually a bit divided into two clades. Some of our methods indicated that they should be recognized as different species, while other outputs indicated that they were one species with two distinct populations. So further studies are needed to determine the taxonomic classification here, but we definitely see a distinct Western river lamprey population in the Central Valley that is distinct from the North Coast population and other Western river lamprey in Oregon and Washington. On a more exciting note, the rest of these different colored lineages really do clearly are very highly supported by all of our analyses as distinct species, including populations in Alameda Creek and the Napa River, shown in purple and deep red here. And those populations had never been reported on before. So these are two novel species of lamprey that no one had ever observed before, which was super exciting. We also see a distinct lineage in the Russian River, in Paynes Creek, and Clear Lake. And those were all reported in Boguski et al. 2012, the study I mentioned a moment ago. So again, we see this really high genetic differentiation in Lampetra, despite only recognizing two species taxonomically at this point. In Entosphenus, the currently recognized species are Pacific lamprey, Pit Klamath Brook lamprey, and Klamath River lamprey, shown here in gray, yellow, and orange on the tree. And molecular species delimitation was attempted in this genus, but the programs performed very poorly and were unable to delimit these currently recognized species, most likely due to a very high sequence similarity. So to simplify that tree a little bit, this is the pattern that we observed, where we did see distinct lineages for these species known as pit Klamath Brook lamprey and Klamath River lamprey. But we do see these two clades of Pacific lamprey kind of confounding the distinction of these other lineages. And the genetic differentiation that we observed is less than we would expect between species level lineages. They sort of look a little bit more like distinct populations. So last, we wanted to see how this diversity is distributed across the landscape. And one question we were particularly interested in is whether the patterns that we see in lamprey is similar to those in other native fish taxa. So these are zoogeographic provinces that were based on patterns of native fish diversity. These were established by Peter Moyle in 2002. So if we overlay our results on top of that map, we see that in Lampetra, we do see this similar pattern to other native fish taxa, where we see distinct populations in the lower Klamath and the North Coast, but there are multiple distinct lineages within the Central Valley subprovince. So this just really indicates that unlike other fish, lamprey are actually diversifying within one subprovince, which is not too shocking since it is the largest subprovince. But this shows that this genus, at least, is following a pattern that's a little bit different than other fish in the Central Valley. In Entus venus, again, the pattern we saw closely follows the pattern of other native fish taxa. There is no distinct lineage in the North Coast subprovince, but we do see these distinct lineages in the Lower Klamath and the Pitt River and Goose Lake subprovinces. So in conclusion, we see really high lamprey diversity in California. Multiple species co-occur at many sites, so getting that species ID correct is really important. Again, in Lampetra, we see this really high genetic differentiation, whereas in the other genus, there's barely any genetic differentiation. And current taxonomy is not sufficient to describe all the diversity that we have here. And again, the geographic distribution of diversity is similar to other native fish taxa. So what's next for me and lamprey? I am applying high throughput genomic methods to see if these patterns we've observed using this method are consistent across the genome. So we used one gene for this study, and it has a lot of really it can tell us a lot, but to get at more fine scale patterns, genomic studies are going to be really helpful. Additionally, I'm developing a species ID and genetic monitoring panel for lamprey in California. And I plan to investigate the genomic basis of life history variation. So I want to know whether there are genes or genomic regions associated with migration versus freshwater resident life histories, and parasitism versus non-parasitism.
Hey, Grace,
So with
just
that,
looking at time. Oh, great. Thank you so much. We do have 1 minute left, and then there is a question in the chat. Perhaps if you could look at that and we can wrap up.
How did I do? So the question in the chat is, how did you do the sequencing? Did you have access to a sequencer, or did you send samples off to a lab? And do you plan to use this research to try and inform or change any fishing or restoration policies at the state level? To answer the first question, we did send these samples off to a sequencing lab. And I do hope that this research will be able to change policies. Mostly, a good first step would be to move forward with formally describing these species so that they are recognized. And beyond that, I would hope that with recognition, these species will be eligible for protection.
Thank you so much, Grace. And I know we were at time. I want to be respectful of everyone's time. If you have any more questions for Grace, please feel free to reach out to her. I'm sure she had her contact information up, but you could probably add that to the chat if needed. But thank you everyone for joining. And again, next week we have our final presentation for the remaining graduate research fellows. So please tune in Tuesday, 2 p.m. Thank you very much. Have a great rest of your day.